Welcome back, everyone. We're about to take a deep dive into a really fascinating case study, all about a drug that made it through the preclinical phase like a champ and snagged that coveted IND, the investigational new drug
Oh,
application.
yeah, the golden ticket to clinical trials.
Exactly. But it's no easy feat. So we're going to try to break down their secret sauce.
Mhm.
What strategies help them clear all those hurdles, and what can we learn from their success? Because getting a drug approved, well, let's just say it takes years, sometimes even a whole decade, and can cost a small fortune.
Absolutely. It's a long and winding road.
It really is. So to help us navigate this journey, we've got a mountain of research reports, all that good stuff, even some firsthand accounts from the scientists involved. And trust me, it's a pretty wild ride.
I bet. I'm already hooked.
You and me both. And get this drug we're diving into, it actually targets a really common condition, the
Ah.
kind of thing that probably affects someone you know,
Mhm.
but actually getting it to work effectively in the body. That was a whole other story, a real nail biter.
Can't wait to hear more.
Oh, you will. But before we get into the nitty-gritty of this particular case study, let's take a step back and look at the big picture of drug development itself. When you think about how a new drug is born, like from scratch, what comes to mind?
Well, the first thing that pops into my head is that initial discovery phase. Scientists are in the lab, like master chefs, cooking up new molecules, testing them out to see if they have any therapeutic potential. It's a lot of trial and error, like throwing spaghetti at the wall and seeing what sticks.
Uh-huh. I love that analogy. And once they find a promising candidate, it's like, okay, now what? How do we actually get this thing into the body where it needs to be?
Exactly. That's when you start thinking about things like should it be a pill, a liquid, maybe an injection? And then there's the whole question of dosage. How much should we give and how often? It's like designing a delivery system, making sure the drug reaches the right place in the body at the right concentration. You don't want to overload the system.
Right. And that's where bioavailability comes into play. It's all about how much of the drug actually makes it into the bloodstream where it can do its job.
You got it. Bioavailability is key. It's like making sure your car has enough fuel to reach its destination.
So can you break down for us why bioavailability is so important? Like what makes it such a big deal?
Well, imagine you're trying to water a plant, but instead of pouring the water directly on the roots, you just dump it on the ground next to it. Is that going to be effective? Probably not. The plant needs that water to reach its roots to actually thrive.
Makes sense.
And it's the same thing with drugs. They got to get into the bloodstream, that highway system in the body to circulate and reach their target. Otherwise, they're just kind of hanging out, not doing much good.
Ah, so they need to hitch a ride on the bloodstream express.
Ha! Exactly. And here's where things get even more interesting. Some drugs have what we call a narrow therapeutic range. This basically means that the difference between a safe and effective dose and a potentially harmful dose, well, it's tiny, like walking a tightrope.
Oh, wow. So the margin for error is really slim.
You got it. It's a delicate balance. You got to be super precise.
Gotcha. So can you give us a real-world example of a drug that has this narrow therapeutic range, to help us understand why getting the dosage right is so critical?
Absolutely. Let's take digoxin, for example. It's a medication used to treat heart failure, a very serious condition.
Okay, I've heard of that one.
So if the blood concentration of digoxin is too low, it's not going to be effective in regulating that heart rhythm. But if it's too high, it can actually backfire and become toxic, which can lead to some really serious complications, even life-threatening ones.
Wow, so it's a real balancing act.
For sure. And that's why bioavailability and getting that dosage right are absolutely crucial, especially for these medications with a narrow therapeutic range.
It's like literally a matter of life and death.
Oh, you said it.
And speaking of life-saving drugs, let's circle back to our case study star. Can you set the stage for us? What kind of drug are we dealing with here, and what makes this case such a success story?
So, without spilling all the beans just yet, this drug we're talking about, it targets a condition that affects millions of people worldwide, a real widespread issue. And what makes this case study so remarkable is that the researchers hit a major roadblock right out of the gate.
A roadblock?
Yeah, they knew the drug had potential, like a diamond in the rough, but getting it to actually work effectively in the body, that proved to be a real challenge. It's like having a car with a powerful engine but flat tires.
So all that power, but nowhere to go.
Exactly.
Okay, so now you've got me really curious. What were some of the obstacles they ran into? And how did they use their knowledge of pharmacokinetics, that whole science of how drugs move through the body, to overcome them?
Well, one of the biggest challenges they faced was figuring out how to get the drug to stay in the body long enough to actually have a therapeutic effect. It was being eliminated way too quickly, like a flash in the pan.
Oh, so it wasn't sticking around long enough to do its job.
Ah, you got it.
Right.
Which meant it wasn't reaching its target in the body at a high enough concentration to really make a difference. It's like trying to put out a fire with a water gun.
Right. You need a steady stream, not just a quick spritz.
Exactly.
Yeah.
So essentially, they had a bioavailability issue on their hands. The drug just wasn't hanging out in the bloodstream long enough to work its magic.
So how did they fix that? Did they change the drug's recipe, the formulation, or maybe find a different way to give it?
They actually explored both of those avenues. But before they could start tinkering with different formulations or delivery methods, they knew they had to get to the bottom of why this drug was vanishing so quickly in the first place. It was like solving a mystery. They had to put on their detective hats and dig deeper into the drug's pharmacokinetic properties.
So they needed to become drug detectives. I love it.
Yeah.
Where did they even begin? What kind of clues were they looking for?
That's a great way to put it. They needed to understand the drug's journey through the body, how it was absorbed, distributed, metabolized, and finally eliminated. We call it ADME for short.
ADME, got it.
And the cool thing is, each step in this ADME journey can be measured and quantified. It's like having a GPS tracker on the drug so you can see exactly where it goes and how long it stays there. And that helps us predict how the drug will behave in the body and ultimately how to optimize its delivery.
Okay, so by understanding this ADME profile, they could basically identify the weak link, the step in the process that was causing this rapid elimination.
Exactly, like finding the leak in a pipe.
But how do you actually measure these things? It sounds pretty complex.
Well, they started by focusing on a few key parameters. One of these was the apparent volume of distribution, or V for short.
Okay, V, I'm listening.
Now, V is a bit tricky because it's not a real physical volume, like the amount of liquid in a glass. It's more of a theoretical concept that reflects how widely a drug spreads throughout the body's tissues, like how much territory it covers.
So if a drug has a high V, does that mean it's distributed more widely throughout the body? And how does that affect how quickly it's eliminated?
You're right on track. Imagine you add a drop of food coloring to a small glass of water. The color spreads quickly, right?
It mixes right in.
But if you add that same drop of food coloring to a giant swimming pool, the color is going to be way more diluted and take much longer to spread.
Ah, I see. So a drug with a high V is like that drop of food coloring in the swimming pool. It's spread out over a larger area, which might mean it takes longer to clear out of the body.
You got it. And the researchers had a hunch that this particular drug might have a relatively high V because of its chemical structure. It had certain properties that made it more likely to enter tissues, kind of like a key fitting into a lock.
Interesting. So that's one piece of the puzzle, this apparent volume of distribution. What other parameters were they looking at? And how did this information ultimately help them solve that problem of the drug disappearing too fast?
Well, another crucial piece of the puzzle was the elimination half-life, which is basically the time it takes for the amount of drug in the body to decrease by half. They needed to figure out how long this drug was actually sticking around in the bloodstream before it got eliminated.
Got it. The elimination half-life.
Yeah.
So if a drug has a short half-life, it means it's being eliminated quickly.
Right.
And that could explain why they were having trouble keeping a steady level of the drug in the bloodstream.
Exactly. And that's precisely what they found. This drug had a surprisingly short half-life, which confirmed their suspicions.
Uh-huh. The mystery deepens.
It does.
Very.
And now, armed with this knowledge, they could finally start brainstorming solutions. Should they try to tweak the drug's structure to make it last longer, like giving it a sturdier backpack? Or maybe find a way to deliver it more frequently to keep a steady supply in the bloodstream?
So it's like choosing between a longer-lasting ice cream cone or eating smaller scoops more often to keep up with the melting. Both strategies could work, but they each have their own pros and cons.
Right. And in this case, modifying the drug structure wasn't really a feasible option. It would have been super time-consuming and expensive, and there was no guarantee it would even work. So they decided to focus their energy on optimizing the delivery method and figuring out the best dosage regimen.
Okay, so they went with the smaller scoops more often approach.
Yeah.
How did they go about fine-tuning that? I'm all ears.
Well, remember how we talked about the importance of bioavailability, getting that drug into the bloodstream effectively?
Like making sure the plant actually gets the water.
Exactly. So they had to consider different routes of administration. Could they maybe bypass the digestive system where some of the drug might get lost or broken down?
Smart. It's like finding a more direct route to that sandcastle on the beach. Did they consider injections?
You got it. They started experimenting with intravenous administration, which means delivering the drug straight into the bloodstream.
Intravenous, right to the source.
Exactly. And this helped them maximize bioavailability and avoid that first-pass metabolism in the liver, where a big chunk of the drug was getting broken down before it could even reach the bloodstream.
The liver's like a bouncer at a club sometimes. It's a little too good at its job.
Uh-huh, that's a good one.
So by delivering it intravenously, they basically snuck the drug past the bouncer. They were able to increase the amount that actually got into the bloodstream. But what about how often they gave it? Did they still need to give it more frequently because of that short half-life?
That's where that elimination half-life data really came in handy. They used that information along with data on how well the drug was working in preclinical models to calculate the optimal dosing interval. They had to find that sweet spot, giving it often enough to maintain a good therapeutic effect, but not so often that it became a burden for patients or caused too many side effects.
It's like finding the perfect rhythm for watering that plant. Not too much, not too little, but just the right amount at the right time to help it flourish.
I like that analogy. And through a lot of careful experimentation and analysis, they were able to figure out a dosing regimen that maintained a good, consistent therapeutic concentration in the bloodstream.
So they finally cracked the code. They took that drug that was sprinting out of the body
Yeah.
and figured out how to make it hang out long enough to do its job.
Exactly. They turned that initial hurdle of rapid elimination into a success story.
This is amazing. They really turned things around. But we know there's more to developing a drug than just understanding the pharmacokinetics, right?
Oh, absolutely.
They also had to play by the rules, follow the regulations and guidelines, right? Can you tell us a bit more about that side of things?
For sure. The FDA, the Food and Drug Administration, they play a huge role in making sure that new drugs are both safe and effective.
Right, they're like the guardians of drug safety.
Exactly. And they have these strict guidelines that researchers have to follow every step of the way, from the initial discovery phase all the way to clinical trials.
So it's like having a set of blueprints for building a house. You can't just start putting up walls without making sure they meet code.
Exactly. And one of the key guidelines that came into play in this case study is known as Good Laboratory Practices, or GLP for short.
GLP, got it.
And these guidelines ensure that all preclinical studies are done with the highest standards of quality and integrity. You want to make sure the data you're basing these important decisions on is solid, right?
Makes sense.
Hmm.
You wouldn't want to build a house on a shaky foundation. So what kinds of things does GLP cover?
Well, pretty much everything from making sure the scientists conducting the research are properly trained and qualified, to the calibration and maintenance of equipment, how samples are handled and stored, and even how the results are documented and reported.
Wow, so it's a really comprehensive system. It's like having a checklist to make sure everything's done by the book.
Exactly.
Yeah.
And the researchers in this case study meticulously followed GLP principles throughout their preclinical work.
They dotted their I's and crossed their T's.
Yes. And this not only ensured the quality of their data, but it also helped to streamline the whole IND submission process.
Smart move. So it sounds like they really did their homework. They not only uncovered the drug's pharmacokinetic secrets, but also made sure their research met the highest standards.
They were thorough. And you know what else is worth noting? They didn't just reinvent the wheel, they actually drew a lot of inspiration and knowledge from the existing scientific literature.
Ah, so they weren't afraid to stand on the shoulders of giants.
Exactly. They tapped into the collective wisdom of researchers who had tackled similar challenges before.
The power of collaboration.
For sure. Can you give us some examples of how they incorporated existing knowledge into their strategy?
Absolutely. One great example is how they used their understanding of the relationship between a drug's lipophilicity and its apparent volume of distribution. Remember how we talked about how a lipophilic drug, one that likes to mingle with fats, is more likely to slip into tissues and have a larger apparent volume of distribution?
Yes. Like that food coloring spreading out in the swimming pool.
Exactly. Well, the researchers knew, based on the drug's chemical structure, that it was pretty lipophilic, a bit of a fat lover. So they anticipated a larger apparent volume of distribution and factored that into their dosage calculations right from the start.
That's smart. They used what they already knew to anticipate potential challenges and adjust their game plan accordingly.
It's like checking the traffic report before you head out on a road trip.
Exactly. And they also looked at previous research on similar drugs to get a feel for the usual metabolic pathways involved, how the body breaks down the drug. They knew that certain enzymes in the liver, the cytochrome P450 enzymes, are like the demolition crew for many drugs.
Ah, the liver, always working overtime. So they were trying to figure out how their drug might be broken down by these enzymes.
Exactly. And by understanding these metabolic pathways, they could predict potential drug interactions. For example, if their drug happened to be metabolized by the same enzyme as another common medication, there could be competition for that enzyme, leading to some unpredictable changes in drug levels.
It's like two people trying to use the same taxi at rush hour.
Uh-huh, a perfect analogy. And this foresight allowed them to design their preclinical studies in a way that would give them valuable information about the drug's metabolic profile and any potential for interactions.
They were thinking several steps ahead. It's like playing chess.
Exactly. And all of this meticulous planning and research really paid off. They successfully navigated the preclinical phase and submitted a really strong IND application, which paved the way for those clinical trials in humans.
Wow, what an incredible journey. They took a potential setback and turned it into a triumph. It really shows the power of scientific ingenuity and a deep understanding of pharmacokinetics.
I agree, and I think their success holds some valuable lessons for the future of drug development. Maybe this kind of meticulous attention to detail will become the new gold standard.
It's exciting to think about the possibilities, especially for drugs with that narrow therapeutic range we discussed earlier. Being able to fine-tune the dosage based on an individual's unique pharmacokinetic profile could make all the difference.
Right, it's like having a custom-made suit versus one off the rack. It just fits better.
A perfect fit. And, this case study also highlights something else that's really important, the value of collaboration and learning from the existing body of scientific knowledge.
Absolutely.
They didn't just work in isolation. They tapped into the collective wisdom of researchers who came before them, building on their successes and learning from their mistakes.
It's the beauty of the scientific process, right? Always building on what we know to push the boundaries further.
Exactly. It's like a relay race, each generation passing the baton to the next.
Love that analogy. And this case study is a perfect example of how even when you face seemingly insurmountable challenges, a combination of ingenuity, careful research, and collaboration can lead to some amazing breakthroughs.
This has been a truly fascinating deep dive. I feel like I've gained a whole new appreciation for the intricate world of drug development and the crucial role that pharmacokinetics plays.
It's been a pleasure exploring this case study with you. I hope our listeners are feeling inspired, knowing that scientists are working tirelessly to develop safer and more effective treatments.
It's a testament to the power of human curiosity, our never-ending quest to understand the world around us, and, in this case, how drugs work in the body.
And that quest continues, always pushing forward, always searching for answers.
And on that note, we'll wrap up this part of our deep dive. But don't worry, we'll be back soon to continue the journey and explore the next chapter in this preclinical success story. Until then, stay curious, keep exploring, and as always, thanks for joining us on the deep dive.
Right. And in this case, modifying the drug structure, it just wasn't really a practical option. It would have been way too time-consuming, super expensive, and honestly, there was no guarantee that it would even work.
Yeah, talk about a gamble.
Totally. So they decided to focus their efforts on optimizing that delivery method, and figuring out the best possible dosage regimen.
Okay, so they went with the smaller scoops more often strategy, right? But how did they actually fine-tune that?
Well, you remember how we were talking about the importance of bioavailability, getting that drug into the bloodstream as effectively as possible?
Right, like making sure the plant actually gets the water, not just watering the dirt around it.
Exactly.
Right.
So they knew they had to consider different routes of administration. Could they maybe find a way to bypass the digestive system, where some of the drug might be lost or broken down before it even gets a chance to work?
Oh, that makes sense, like finding a more direct path, right?
Exactly, like a shortcut. So they started looking into intravenous administration, which means delivering the drug straight into the bloodstream.
Ah, straight to the source.
Exactly, no detours. And this actually helped them maximize that bioavailability, and it also helped them avoid that first-pass metabolism in the liver, which is where a big chunk of the drug was getting broken down before it could even reach the bloodstream.
So they found a way to sneak it past the liver's security checkpoint.
Exactly.
Clever. But what about the frequency? How often they gave the drug? Did they still need to give it more often because of that short half-life we talked about?
Well, that's where the elimination half-life data really came in handy. They used that information, along with data on how well the drug was actually working in preclinical models, those animal studies, to calculate the optimal dosing interval. They had to find that sweet spot, giving it often enough to maintain a good therapeutic effect, but not so often that it became a real burden for the patients or, started to cause a bunch of side
Right, you
effects.
don't want to go overboard. So it's like finding the perfect rhythm for watering that plant, not too much, not too little, but just the right amount at the right time to help it really thrive.
I like that analogy. It's all about balance, right?
Hmm.
And through a lot of careful experimentation and analysis, they were finally able to figure out a dosing regimen that kept a consistent therapeutic level of the drug in the bloodstream.
So they finally cracked the code. They took that drug that was practically sprinting out of the body and figured out how to make it hang out long enough to actually do its job.
That's awesome.
It is. They turned that initial hurdle of rapid elimination into a real success story.
I love it when a plan comes together. But we know there's more to developing a drug than just understanding those pharmacokinetic principles, right? They also had to navigate that whole world of regulations and
Oh,
guidelines.
for sure.
Can you tell us a bit more about that aspect of things?
Absolutely. So the FDA, the Food and Drug Administration, they have a really important job. They're the ones who make sure new drugs are both safe and effective for people to use.
They're like the gatekeepers, right? Making sure everything is up to par.
Exactly.
Sure.
And they have these really strict guidelines that researchers have to follow every step of the way, from the very beginning when they're first discovering a drug, all the way to those clinical trials in humans.
Right. So it's like having a set of blueprints for building a house. You can't just start putting up walls without making sure they meet code. Exactly. And one of the key guidelines that was really important in this case study is known as good laboratory practices, or GLP.
GLP, got it. And what do those guidelines do?
Well, they're basically there to make sure that all those preclinical studies are conducted with the highest possible standards, ensuring quality and integrity in every step of the research process. You don't want to base important decisions about a drug's safety and effectiveness on sloppy or unreliable data, right?
Makes total sense. GI go, right.
Yeah.
Garbage in, garbage out.
Yeah.
So what kind of things does GLP cover?
Well, pretty much everything. I mean, it covers the training and qualifications of the scientists doing the research, the calibration and maintenance of all the equipment they're using, the handling and storage of those precious samples, and even how they document and report all their findings. It's really comprehensive.
Wow, so it's like a super detailed checklist, making sure every I is dotted and every S is crossed.
You got it. And in this case study, the researchers were super meticulous about following those GLP principles throughout their entire preclinical process.
So they really did their due diligence.
They did. And that not only made sure their data was top-notch, but it also helped to make that whole IND submission process a lot smoother, when they were applying to get permission to start clinical trials in people.
Ah, so it's like having all your paperwork in order when you go through customs.
Exactly. And another thing that's really important to mention is that they didn't just reinvent the wheel. They actually drew a lot of inspiration and knowledge from the existing body of scientific research, from all the work that had been done before them.
Ah, the power of collaboration and learning from others.
Exactly. They were smart enough to tap into the collective wisdom of all those researchers who had already tackled similar challenges, building on their successes and learning from their mistakes.
Like it's saying standing on the shoulders of giants, right?
Exactly. And that's what's so cool about science. It's this constantly evolving process of discovery, always building on what we already know to push the boundaries further.
I love that. Could you give us some specific examples of how they incorporated that existing knowledge into their strategy?
Absolutely. One great example is how they applied their understanding of the relationship between a drug's lipophilicity and its apparent volume of distribution. Remember how we talked about how those lipophilic drugs, the ones that love to dissolve in fats, are more likely to enter tissues and end up with a larger apparent volume of distribution?
Yeah, it's like those oil and water don't mix kind of thing, right?
Exactly. Well, the researchers in this case study, they already knew just by looking at the chemical structure of their drug that it was pretty lipophilic. So they anticipated that it would have a larger apparent volume of distribution, and they actually factored that into their dosage calculations right from the get-go.
Smart. So they use that existing knowledge to sort of anticipate potential challenges and adjust their strategy accordingly. It's like checking the traffic report before you head out on a road trip, to avoid any major delays.
Exactly. And they also looked back at previous research on similar drugs, just to get a sense of what those typical metabolic pathways were, how the body usually breaks down those types of drugs. And they knew that certain enzymes in the liver, those cytochrome P450 enzymes, they play a huge role in metabolizing a lot of different drugs.
The liver, always working hard to detoxify our systems.
Right. And by understanding these metabolic pathways, they could actually predict predict potential drug interactions. For example, if their drug happened to be metabolized by the same enzyme as another commonly used medication, there could be some competition for that enzyme, which could lead to some unpredictable and potentially dangerous changes in those drug levels.
So they were thinking ahead, trying to anticipate any potential roadblocks.
Exactly, like playing a game of chess, always thinking a few moves ahead. And all of this foresight actually allowed them to design their preclinical studies in a way that would give them valuable data about the drug's metabolic profile and its potential for any unwanted interactions.
They were really thorough. It sounds like they left no stone unturned. And all that meticulous data gathering, the careful analysis, and using all that existing knowledge, it all paid off in the end, right?
It did. They successfully navigated that whole preclinical phase and were able to submit a really strong IND application, paving the way for those clinical trials in humans.
Amazing. So they took what could have been a major setback and transformed it into a total triumph. It really goes to show you the power of scientific ingenuity and a deep understanding of how drugs interact with the body.
I completely agree. And I think their success story holds some really valuable lessons for the future of drug development. Who knows, maybe this kind of meticulous attention to detail will become the new gold standard in the field.
It's an exciting thought. And it makes you wonder, how will this approach shape the future of drug development? Do you think this kind of focus on personalized medicine, tailoring treatments to an individual's specific needs, is where the field is headed?
Well, I definitely think it has the potential to revolutionize the way we develop new drugs. Imagine a future where we're not just throwing darts in the dark, hoping to hit the target. Instead, we're using these principles to design drugs that are truly tailored to each patient's unique needs and characteristics.
So it's like moving away from that one-size-fits-all approach to something much more precise and individualized.
Exactly. And this case study shows us that it's not just some futuristic fantasy. It's actually achievable with the right tools and knowledge. And it could be especially impactful for those drugs with a narrow therapeutic index, the ones where that margin for error is so tiny.
Right, like that digoxin example we talked about earlier.
Exactly. Being able to really fine-tune the dosage based on a person's individual pharmacokinetic profile could make a huge difference in how well their treatment works and also help minimize the risk of those potentially dangerous side effects.
So it's safer and more effective. A win-win situation. And, this case study also highlights another really important point, the value of collaboration and learning from all that existing scientific knowledge that's out there.
Absolutely. Building on the work of those who came before us.
Exactly. It's like standing on the shoulders of giants, right?
Mm-hmm.
Using the accumulated knowledge and insights from the past to help us make even greater strides in the future.
Exactly. And that's what makes science so powerful. It's a constantly evolving process of discovery, always building upon the work that's been done before.
It's like we're all part of this incredible relay race, each generation passing the baton to the next. And this case study really showcases how, even when faced with what seem like insurmountable obstacles, that combination of human ingenuity, meticulous research, and a willingness to collaborate, it can lead to some pretty remarkable breakthroughs.
I couldn't agree more. It's a testament to the power of human curiosity and our relentless pursuit of knowledge.
I completely agree. And I think their success story holds some really valuable lessons for the future of drug development. Who knows, maybe this kind of meticulous attention to detail will become the new gold standard in the field.
It's an exciting thought. And it makes you wonder, how could this approach shape the future of drug development? Do you think this kind of focus on personalized medicine, tailoring treatments to an individual's specific needs is where the field is headed?
Well, I definitely think it has the potential to revolutionize the way we develop new drugs. Imagine a future where we're not just throwing darts in the dark, hoping to hit the target. Instead, we're using these principles to design drugs that are truly tailored to each patient's unique needs and characteristics.
So it's like moving away from that one-size-fits-all approach to something much more precise and individualized. Exactly. And this case study shows us that it's not just some futuristic fantasy. It's actually achievable with the right tools and knowledge. And it could be especially impactful for those drugs with a narrow therapeutic index, the ones where that margin for error is so tiny.
Right, like that digoxin example we talked about earlier.
Exactly. Being able to really fine-tune the dosage based on a person's individual pharmacokinetic profile could make a huge difference in how well their treatment works and also help minimize the risk of those potentially dangerous side effects.
So it's safer and more effective, a win-win situation. And this case study also highlights another really important point, the value of collaboration and learning from all that existing scientific knowledge that's out there.
Absolutely. Building on the work of those who came before us.
Exactly. It's like standing on the shoulders of giants, right?
Yeah.
Using the accumulated knowledge and insights from the past to help us make even greater strides in the future.
Exactly. And that's what makes science so powerful. It's a constantly evolving process of discovery, always building upon the work that's been done before.
It's like we're all part of this incredible relay race, each generation passing the baton to the next. And this case study really showcases how, even when faced with what seem like insurmountable obstacles, that combination of human ingenuity, meticulous research, and a willingness to collaborate, it can lead to some pretty remarkable breakthroughs.
I couldn't agree more. It's a testament to the power of human curiosity and our relentless pursuit of knowledge.
It's really inspiring to see what's possible when brilliant minds come together with a shared goal. This deep dive has been an incredible journey, and I want to thank you for sharing your expertise with us today.
It's been my pleasure.
I think our listeners will walk away from this episode with a whole new appreciation for the complex world of drug development, all the hard work and dedication that goes into bringing those life-changing treatments to the people who need them.
I hope so.
And, it really underscores the importance of supporting scientific research and encouraging those future generations of scientists who will continue to push the boundaries of what's possible. To all our listeners out there, keep those questions coming, stay curious, and never stop exploring. And that's a wrap for today's deep dive. Thanks for joining us.